So now we are starting the chapter 9, that is Biomolecules. ये जो सीरीज़ है ना, ये रिवीजन सीरीज़ है। इसमें आप स्टार्टिंग से अगर पढ़ना चाहते हो तो ये पढ़ने के लिए नहीं है। जिनने NCERT पढ़ रखी है बस उनको रिवीजन करना हो, लाइंस माइंड में रखनी हो, तो उसके लिए ये सीरीज़ बहुत हेल्पफुल है। आप ये पॉडकास्ट उसके लिए सुन सकते हो। So let's proceed. Chapter 9, Biomolecules. There is a wide diversity in living organisms in our biosphere. Now a question that arises in our minds is, are all living organisms made up of the same chemicals, that is elements and compounds? You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue or a microbial pest, we obtain a list of elements like carbon, hydrogen, oxygen and several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust as an example of non-living matter, we obtain a similar list. What are the differences in between these two lists? In absolute terms, no such differences could be made out. In absolute terms, no such differences could be made out. All the elements present in a sample of earth crust are also present in a sample of a living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in earth crust. ये जो लाइन स्पीड में और फास्ट जो पढ़ रहा हूँ ना मैं, मीन्स इतना इम्पोर्टेंस नहीं है, मीन्स ऐसे चैप्टर का इंट्रोडक्शन है, तो इसलिए उसमें टाइम वेस्ट नहीं करते. So let's know how to analyze chemical composition. We can continue asking in the same way, what type of organic compounds are found in living organisms? How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc. and grind it in a trichloroacetic acid, Cl3CCOOH, using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called filtrate, or more technically the acid soluble pool, and the second the retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in the acid soluble pool. In higher classes, we will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extracts the compound, then subject the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical technique, when applied to the compound, give us an idea of the molecular formula and the probable structure of the compound. All the carbon compound that we get from living tissue can be called as biomolecule. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weighs a small amount of a living tissue, say a leaf or liver, and this is called weight, and dry it. All the water evaporates. The remaining material gives us dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is CO2 and water vapors, and are removed. What is remaining is called ash, means rock. This ash contains inorganic elements like calcium, magnesium, etc. Organic नहीं, inorganic element. ठीक है, ऐसे confuse करिए, like calcium and magnesium. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid soluble fraction. Therefore, elemental analysis gives elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds give an idea of the kind of organic and inorganic constituents in the table 9.2. We will read the chapter later, present in living tissues. From a chemistry point of view, one can identify functional groups like aldehyde, ketone, aromatic compound, etc. But from a biological point of view, we shall classify them into amino acid, nucleotides, bases, fatty acids, etc. I am going to read the table 9.1, a comparison of elements present in non-living and living matter. Element is hydrogen and percent weight of earth crust in human body. Percent weight of earth crust, hydrogen is 0.14 and in human body is 0.5, and percent carbon in earth crust is 0.03 and in human body is 18.5, and oxygen in earth crust is 46.6 and in human body is 65.0. Nitrogen in earth crust is very little and in human body is 3.3. Sulfur in earth crust is 0.03 and in human body is 0.3. Sodium in earth crust is 2.8 and in human body is 0.2. Calcium in earth crust is 3.6 and in human body is 0.2. Calcium in earth crust is 3.6 and in human body is 1.5. Magnesium in earth crust is 2.1 and in human body is 0.1. Silicon in earth crust is 27.7 and in human body is negligible. List of representative inorganic constituent of living tissue. Component name is sodium, formula is Na upper positive, potassium is K upper positive, calcium is Ca double plus, magnesium is Mg double plus, water is H2O, compounds are NaCl, CaCO3, PO4 3 negative, SO4 2 negative. Now next is amino acids are organic compounds containing an amino group and an acidic group, a substituent on the same carbon, that is the alpha carbon. Hence they are called alpha amino acids. They are substituted methanes. There are 4 substituent groups occupying the 4 valency positions. These are hydrogen, carboxyl group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. In protein, which occur are of only 20 types. The R group in this proteinaceous amino acid could be hydrogen. The amino acid is called glycine. If the hydrogen is there, the amino acid is called glycine. And in methyl group, it is called alanine. Hydroxyl methyl, it is serine, etc. 3 of the 20 are shown in the figure 9.1. The chemical and physical properties of amino acids are essentially of the amino, carboxyl and R functional groups. Based on number of amino and carboxyl groups, there are acidic, example glutamic acid, basic, that is lysine, and neutral, that is valine, which you can remember by NV, means neutral valine amino acid, NV sir. Okay. Similarly, there are aromatic amino acids like tyrosine, phenylalanine, tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH groups. Hence, in solutions of different pH, the structure of amino acid changes. Understood? Lipids are generally water insoluble. यहाँ पे चेंज करके soluble कर सकता है, but correct है insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl, that is CH3, or ethyl C2H5, or higher number of CH2 group, 1 carbon to 19 carbon. For example, palmitic acid has 16 carbons including carboxyl carbon. Palmitic has 16 including carboxyl carbon. Arachidonic acid has 20 carbon including the carboxyl carbon. Arachidonic 20, palmitic 16 including carboxyl carbon. ठीक है, इंक्लूडिंग एक्सक्लूडिंग का लफड़ा होगा बट कर लेना तुम, ठीक है। इंक्लूडिंग के साथ है, पाल्मेटिक में 16, आर्किडोनिक में 20। हो गया माइंड में सेट। चलो आगे बढ़ते हैं। फैटी एसिड कुड बी सैचुरेटेड विदाउट डबल बॉन्ड और अनसैचुरेटेड विथ 1 or more C double bond C. Another simple lipid is glycerol, which is trihydroxypropane. Glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglyceride, diglyceride and triglycerides. These are also called fats and oils based on melting points. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Can you identify a fat from the market? Some lipids have phosphorus and a phosphorylated organic compound in them. Some lipids have phosphorus and a phosphorylated organic compound in them. These are phospholipids. Understood? They are found in cell membrane. Lecithin is one example, which is a phospholipid. Some tissues, especially the neural tissue, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases, adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acid are nucleotides. Nucleic acids like DNA and RNA consist of nucleotides only. Nucleic acid like DNA and RNA consist of nucleotides only. Only है यहाँ पे, ठीक है, important line है, only correct है. DNA and RNA function as genetic material. अब ये यहाँ पे पेज 107 पे कुछ स्ट्रक्चर्स दिए हुए हैं, ये आप देख लेना, ठीक है। हाइड्रोजन है तो ग्लाइसिन, CH3 मिथाइल है तो अलानिन, CH2OH है तो सेरिन, यानी कि मिथाइल हाइड्रोक्साइल है तो, ठीक है, हाइड्रोक्सी मिथाइल है तो सेरिन है, ठीक है। फैटी एसिड है पाल्मेटिक एसिड, ग्लाइसरॉल का स्ट्रक्चर दिया हुआ है, फॉस्फोलिपिड, ग्लाइसिन का स्ट्रक्चर दिया हुआ है यहाँ पे, ठीक है। फैट्स एंड ऑयल्स, लिपिड, न्यूक्लियोसाइड, इनका भी स्ट्रक्चर दिया हुआ है, पढ़ सकते हैं, ठीक है। नेक्स्ट इज प्राइमरी एंड सेकेंडरी मेटाबोलाइट्स. द मोस्ट एक्साइटिंग एस्पेक्ट ऑफ chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and if possible synthesizing them. If one were to make a list of molecules, such a list would have thousands of organic compounds including amino acids, sugars, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds shown in the figure 9.1. अब देखना तुम फिगर, ठीक है। These are called primary metabolites shown in the figure 9.1. कौन सा है फिगर 9.1, ठीक है, ये जो दिए हुए हैं. These are called primary metabolites. However, when one analyzes plants, fungal and microbial cells. However, when one analyzes plant, fungal, and microbial cells, one would see thousands of compounds other than this called primary metabolites. Example, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigment, sands, gums, spices. These are called secondary metabolite. Understood? जो बोला अभी एग्जाम्पल वो है सेकेंडरी मेटाबोलाइट्स के. ठीक है, कौन कौन है? alkaloid, flavonoid, rubber, essential oils, antibiotic, colored pigment, sands, gums, spices. While primary metabolites have identifiable functions and play non-role in the normal physiological process, we do not at the moment understand the role or function of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example, rubber, drugs, spices, scents and pigment. यूजफुल जो है ह्यूमन वेलफेयर के लिए वो है रबर, ड्रग, स्पाइसेस, सैंड्स एंड पिगमेंट्स. Some secondary metabolites have ecological importance. In the later chapters and here you will learn more about this. अब टेबल पढ़ रहे हैं अपन सम सेकेंडरी मेटाबोलाइट्स की. पिगमेंट्स में आते हैं कैटिनॉइड्स और एंथोसाइनिन. पिगमेंट में आते हैं कैटिनॉइड और एंथोसाइनिन्स. आंखें बंद करके याद रखना, ठीक है. एल्कलॉइड्स में आते हैं मॉर्फिन एंड कॉडिन एक्सेट्रा. टर्पिनॉइड्स में आते हैं मोनोटर्पिन्स, डाइटर्पिन्स एक्सेट्रा. एसेंशियल ऑयल्स में लेमन ग्रास ऑयल. टॉक्सिन में एब्रिन रेसिन. लेसिटिन्स में कॉन्कावेलिन ए. ड्रग में विनब्लास्टिंग एंड कुर्क्यूमिन एक्सेट्रा. पॉलीमेरिक सब्सटेंस में आते हैं रबर, गम्स, सेलुलोज. Now next we are going to read is biomacromolecule. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weight ranging from 18 to around 800 daltons approximately. The acid insoluble fraction has only 4 types of organic compound, that is protein, nucleic acid, polysaccharide and lipid. एसिड इंसॉल्युबल में कौन से हैं 4 टाइप के ऑर्गेनिक कंपाउंड? प्रोटीन, न्यूक्लिक एसिड, पॉलीसैकेराइड एंड लिपिड. दिस क्लासेस ऑफ कंपाउंड विद द एक्सेप्शन ऑफ लिपिड्स हैव मॉलिक्यूलर वेट इन द रेंज ऑफ 10,000 डाल्टन्स एंड अबव. फॉर दिस वेरी रीजन बायोमोलिक्यूल्स, that is chemical compound found in living organisms, are of 2 types. One, those which have molecular weight less than 1000 Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid insoluble fraction are called macromolecule or biomacromolecules. The molecules in the insoluble fractions, with the exception of the lipids, are polymeric substance. The molecule in the insoluble fraction, with the exception of lipids, are polymeric substance. The molecule in the insoluble fraction, with the exception of lipids, are polymeric substances. The molecule in the insoluble fraction, with the exception of lipid, are polymeric substances. Important line है. Then why do lipids whose molecular weights do not exceed 800 Dalton come under the acid insoluble fraction, that is macromolecular fraction? सोचो why? This is because lipids are indeed small molecular weight compound and are present not only as such but also arranged into structure like cell membrane and other membranes. When we grind tissue, we are disrupting the cell structure. Cell membrane and other membrane are broken into pieces and form vesicles which are not water soluble. Therefore, this membrane fragment in the form of vesicle gets separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. Lipids are not strictly macromolecules. The acid soluble pool represent roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together they represent the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organisms. Table 9.4. चलो ठीक है, component water and next is percent of the total cellular mass. Water का है 70 से 90% of the total cellular mass. Protein का है 10 से 15% of the total cellular mass. Carbohydrate का है 3%, lipid का है 2%, nucleic acid का है 5 से 7%, ions का है 1%. Now again water 70 to 90, protein 10 to 15, carbohydrate 3, lipid 2, न्यूक्लिक एसिड 5 to 7, आयंस 1. आराम से चल रहे हैं ताकि चैप्टर यहीं पे आपको माइंड में फिट हो जाए. ठीक है, कोई दिक्कत नहीं. नेक्स्ट इज प्रोटीन्स. प्रोटीन्स आर पॉलीपेप्टाइड्स. दे आर लीनियर चेन्स ऑफ अमीनो एसिड लिंक्ड बाय पेप्टाइड बॉन्ड्स शोन इन द फिगर 9.3. देखो उठ के. ठीक है, ईच प्रोटीन इज अ पॉलीमर ऑफ अमीनो एसिड्स. As there are 20 types of amino acids, example alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n numbers of times. This information about the amino acid contained is important as later in your nutrition lessons you will learn that certain amino acids are essential for our health and they have to be supplied through our diet. Hence, dietary proteins are the source of essential amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make, while we get essential amino acid through our diet or food. Protein carry out many functions in living organisms. Some transport nutrients across cell membrane, some fight infectious organisms, some are hormones, some are enzymes, etc. Table 9.5 में आप पढ़ेंगे अपन. ठीक है, collagen is the most abundant protein in animal world and ribulose, bisphosphate, carboxylase, oxygenase, rubisco is the most abundant protein in the whole of the biosphere. अब some protein in their function वाली टेबल. ठीक है, प्रोटीन में आता है कोलाजिन. फंक्शन है इंटरसेल्यूलर ग्राउंड सब्सटेंस. इंटर की जगह इंट्रा कर सकते हैं. ठीक है, बट करेक्ट है इंटरसेल्यूलर ग्राउंड सब्सटेंस. ट्रिप्सिन है एंजाइम, फंक्शन है उसका एंजाइम. इंसुलिन है प्रोटीन, फंक्शन है हार्मोन. एंटीबॉडी प्रोटीन, फंक्शन है फाइट इन्फेक्टियस एजेंट. रिसेप्टर है प्रोटीन, फंक्शन है सेंसरी रिसेप्शन, स्मेल, टेस्ट, हार्मोन, एक्सेट्रा. प्रोटीन है GLUT4, फंक्शन है एनेबल ग्लूकोज ट्रांसपोर्ट इनटू द सेल. नेक्स्ट इज पॉलीसैकेराइड्स. The acid insoluble pellet also has a polysaccharide carbohydrate as another class of macromolecule. Polysaccharides are long chain of sugars. They are threads, literally a cotton thread, containing different monosaccharide as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is polymeric polysaccharide consisting of only one type of monosaccharide, example glucose. Only आ गया तो मीन्स इम्पोर्टेंट है. ठीक है, cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Inulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called reducing end and the left end is called non-reducing end. Right R आता है called reducing end RR. ठीक है. Left is non-reducing. It has branches as shown in the figure in the form of cartoon. In figure 9.2, starch form helical secondary structure. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. The starch I2 is blue in color. Cellulose does not contain complex helices and hence cannot hold I2. ठीक है. Plant cell walls are made of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and are chemically modified sugars. Example, glucosamine, N-acetyl, galactosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymer. Chitin is a homopolymer. वाह भाई वाह, सपने देखे बड़े बड़े, बनाना कुछ भी कर ना सके. Next is nucleic acid. The other type of macromolecule that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharide and polypeptide, this comprises the true macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third is a phosphoric acid or phosphate. One is heterocyclic compound, second is monosaccharide, and third is phosphoric acid or phosphate. Understood? So now let's proceed further. As you notice in figure 9.1, the heterocyclic compound in nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines while the rest are substituted pyramids. आग होती है वो प्योर होती है, उससे याद रख सकते हो आप. आग से ए का मतलब एडेनिन, जी से ग्वानिन. The skeletal heterocyclic ring is called as purine and pyrimidines respectively. Understood? The sugar found in polynucleotide is either ribose, a monosaccharide pentose, or 2-deoxyribose. A nucleic acid contains deoxyribose is called deoxyribonucleic acid DNA, while that which contains ribose is ribonucleic acid, that is RNA. Next is structure of proteins. Proteins, as mentioned earlier, are heteropolymer containing strings of amino acid. Structure of molecules means different things in different contexts. In inorganic chemistry, the structure invariably refers to the molecular formula. Example, NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecules while representing the structure of the molecules. Example, benzene, naphthalene, etc. Physics conjure up the three-dimensional view of the molecular structure, while the biologists describe the protein structure at 4 level. The sequence of the amino acid, that is the positional information in a protein, which is first amino acid, which is second, and so on, is called a primary structure of a protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acids. Right end is represented by the last amino acid, means RL. ठीक है, R से right, L से last. The first amino acid is also called as N-terminal amino acid. The last amino acid called C-terminal amino acid. First amino acid is N-terminal. The first amino acid is also called as N-terminal amino acid, and the last amino acid is called the C-terminal amino acid. LC means last is C-terminal. A protein thread does not exist throughout an extended rigid rod. The thread is folded in the form of helix, similar to a revolving staircase. Of course, only some portion of the protein thread are arranged in the form of helix. In proteins, only right-handed helices are observed. Only right-handed helices are observed. Other lines of the protein thread are folded into other forms in what is called the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of protein. Which structure is absolutely necessary for many biological activities of protein? Tertiary. कंफ्यूज करने के लिए ऑप्शन में प्राइमरी, सेकेंडरी, टर्शियरी, क्वाटनरी आएगा. करेक्ट आंसर है टर्शियरी. Some proteins are an assembly of more than one polypeptide. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other, example linear string of spheres, sphere arranged one upon each other in the form of cube or plate, is the architecture of a protein, otherwise called the quaternary structure of a protein. Example, linear string of the sphere arranged one upon each other in the form of cube or plate is the architecture of a protein, otherwise called the quaternary structure of a protein. Adult human hemoglobin consists of 4 subunits. 2 of these are identical to each other. Are identical की जगह not identical कर देगा, but correct है are identical to each other, has 2 subunits of alpha type and 2 subunits of beta type. Together constitute the human hemoglobin, that is Hb. Enzymes. Almost all enzymes are protein. Important line है. Almost all enzymes are protein. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has primary structure, that is amino acid sequence of the protein. An enzyme, like any protein, has the secondary and tertiary structure. When you look at a tertiary structure, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself, and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzyme through their active site catalyze reaction at a high rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention here. In inorganic catalysts inorganic catalysts work efficiently at high temperature and high pressure. Inorganic catalyst work करते हैं high temperature and high pressure पे, while enzyme get denatured. Inorganic catalyst work at high temperature and high pressure, while enzyme get damaged at high temperature, say above 40 degree Celsius. यहाँ से उठ के आ सकती है लाइन, या फिर मल्टीपल चॉइस क्वेश्चन में आ सकता है at a high temperature, that is above 40 degrees Celsius. However, enzyme isolated from organisms who normally live under extremely high temperature, example hot vents and sulfur spring, are stable and retain their catalytic power even at high temperature up to 80 to 90 degrees Celsius. Their thermal stability is thus an important quality of enzyme isolated from thermophilic organism. Next is chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compound undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter. When ice melts into water or when water becomes a vapor. When ice melts into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, BaOH2 plus H2SO4 give BaSO4 plus 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of a physical or chemical process refers to the amount of product formed per unit time. It can be expressed as rate is equal to gamma P upon gamma T, means product upon time. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature, among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10 degree Celsius change in either direction. यहाँ पे ऑप्शन आ जाएंगे अलग अलग, बट करेक्ट है 10 डिग्री चेंज इन आइदर डायरेक्शन, 10 डिग्री सेल्सियस change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed one. When enzyme catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction. When enzyme catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, CO2 plus H2O in the presence of carbonic anhydrase gives H2CO3, that is carbonic acid. In the absence of any enzyme, this reaction is very slow with about 200 molecules of HDCO3, that is carbonic acid, being formed in an hour. However, by using the enzymes present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. How much? 6 lakh every second. यहाँ पे चेंज करके मिनट कर सकता है, बट करेक्ट है सेकंड. The enzyme has accelerated the reaction rate by about 10 million times. The power of the enzyme is incredible indeed. There are thousands of types of enzymes, such each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. Multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzyme, is called a metabolic pathway. For example, glucose gives 2-pyruvic acid. C6H12O6 plus O2 gives 2C3H4O3 plus H2O. For example, is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzymes catalyzed metabolic reaction. When you study respiration in chapter 12, you will study this reaction. At this stage, you should know that this is very metabolic pathway with 1 or 2 additional reactions give rise to a variety of metabolic end products. In our skeletal muscle, under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast during fermentation, the same pathway leads to the production of ethanol, that is alcohol. Hence, in different conditions, different products are possible. How do the enzyme bring about such high rates of chemical conversions? To understand this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. A chemical which is converted into a product is called a substrate. Chemical which is converted into a product is called a substrate. An enzyme, that is protein with three-dimensional structure including an active site, convert a substrate into a product, that is P symbol. Symbolically it can be depicted as substrate gives product. ठीक है? The chemical which is converted into a product is called substrate. ठीक है? Chemical. It is now understood that the substrate has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES complex. E stands for enzyme, enzyme substrate complex. This complex formation is a transient phenomenon. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Very soon after the expected bond making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of product. The pathway of this transformation must go through the so-called transition state structure. So-called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structure states are unstable. All other intermediate structure states are unstable. Stability is something related to energy status of the molecule or the structure. Hence, when we look at it this pictorial through a graph, it looks like something as in figure 9.4, जहाँ पे y एक्सिस पे है पोटेंशियल एनर्जी, x एक्सिस पे है प्रोग्रेस ऑफ रिएक्शन. सबस्ट्रेट प्रोडक्ट में बन रहा है, बीच में ट्रांजिशन स्टेट है. ठीक है, वहाँ पे एक्टिवेशन एनर्जी विदाउट एंजाइम ज़्यादा है, एक्टिवेशन एनर्जी विद एंजाइम कम है. The y-axis represent the potential energy contained, the x-axis represent the progression of the structural transformation or state through the transition state. You would notice two things: the energy level difference between substrate and product. If product is at lower level than substrate, the reaction is an exothermic. नीचे रहेगा तो exothermic. One need not supply energy by heating in order to form the product. However, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction, the substrate has to go through a much higher energy state or transition state. The difference in average energy contained of substrate from that of this transition state is called activation energy. The difference in average energy contained of substrate from that of this transition state is called activation energy. Enzyme eventually bring down this energy barrier, making the transition of substrate to product more easy. Next is nature of enzymes. Nature of enzyme action. Each enzyme has a substrate S binding site in its molecule, so that a highly reactive enzyme substrate complex, ES is formed, where E for enzyme, S for substrate. This complex is short-lived and dissociate into its product and the unchanged enzyme with an intermediate formation of the enzyme product complex. The formation of enzyme substrate complex is essential for catalyst. E plus S, ES का complex, फिर वो बनेगा EP एंजाइम प्रोडक्ट, और फिर वापस दूर हो जाएंगे अलग अलग एंजाइम प्लस प्रोडक्ट अलग अलग. The catalytic cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of the enzyme, fitting into the active site. Understood? First, the substrate binds to the active site of the enzyme, fitting into the active site. Second, the binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Third is the active site of the enzyme, now in close proximity of the substrate, break the chemical bond of the substrate and the new enzyme product complex is formed. And the new enzyme product complex is formed. For this, the enzyme releases the product of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Factor affecting enzyme activity. The activity of an enzyme can be affected by a change in the condition which alter the tertiary structure of the protein. This includes temperature, pH, change in substrate concentration, or binding of specific chemicals that regulates its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH, called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporary inactive state, whereas high temperature destroys enzymatic activities because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, that is Vmax, which is not exceeded by any further rise in concentration of the substrate. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in the concentration of the substrate. This is because the enzyme molecule are fewer than the substrate molecule, and after saturation of this molecule, there are no free enzyme molecule to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shut off enzymatic activity, the process is called inhibition and the chemical is called inhibitor. Understood? जो ग्राफ है यहाँ पे pH और टेम्परेचर का सेम है, मीन्स थोड़ा सा डिफरेंस है, ठीक है, हाफ अंडा, और जो कंसंट्रेशन का है उसमें x एक्सिस पे है Km और y एक्सिस पे है वेलोसिटी ऑफ रिएक्शन Vmax/2, ठीक है, और pH टेम्परेचर है x एक्सिस पे और एंजाइमेटिक एक्टिविटी है y एक्सिस पे, दोनों pH और टेम्परेचर के डायग्राम. अंडरस्टूड? When the binding of a chemical shut off enzymatic activity, the process is called inhibition and the chemical is called inhibitor. Inhibition and inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibit the activity of the enzyme, it is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind and as a result the enzyme action declines. Example, inhibition of succinate dehydrogenase by malonate, which closely resemble the substrate succinate in a structure. यहाँ से क्वेश्चन बहुत आया है, ठीक है, वापस से पढ़ देते. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate. किसके साथ? substrate के साथ compete करता है for the substrate binding site of the enzyme. Consequently, the substrate cannot bind and as a result the enzyme action declines. Example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in its structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Next is classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of reactions they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by a 4-digit number. Oxidoreductase dehydrogenases, enzyme which catalyze oxidoreduction between two substrates S and S-. Example: S reduced plus S- oxidized gives S oxidized and S- reduced. चेंज हो गया, ठीक है, इंटरचेंज. Transferases, enzymes catalyzing a transfer of group G other than hydrogen between a pair of substrate S and S-. Example: S-G plus S- gives S plus S- -G. Hydrolysis, enzyme catalyzing hydrolysis of ester, ether, peptide, glycosidic, C single bond C, C single bond halide or P N bonds. Lysis, enzyme catalyze removal of group from substrate by mechanism other than hydrolysis leaving double bonds is lysis. Hydrolysis का है enzyme catalyzing hydrolysis of ester, ether, peptide, glycosidic, C single bond C, C halide or PN bonds. Lysis, enzyme that catalyze removal of group from the substrate by mechanism other than the hydrolysis leaving double bond is known as lysis. C single bond C दोनों के ऊपर इस पे X है, एक पे Y है, gives X-Y plus C double bond C. Understood? These are lysis. Isomerases includes all enzyme catalyzing interconversion of optical, geometrical or positional isomers. Isomers, important line, includes all enzyme catalyzing interconversion of optical, geometric or positional isomers. Optical, geometric or positional isomers. Ligases, enzyme catalyzing the linking together of two compounds. Example: enzyme which catalyze joining of CO, CS, CN, PO, etc. bonds. Next is cofactor. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituent called cofactor are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. 3 kinds of cofactors may be identified: prosthetic group, coenzymes, and the metalline. Prosthetic group, coenzyme, and the metallions. Prosthetic groups are the organic compound and are distinguished from the other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is a part of the active site of the enzyme. For example, in peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen, heme is the prosthetic group and it is a part of the active site of the enzyme. डबल इसलिए पढ़िए ताकि माइंड में सेट हो जाए, ठीक है. मुतालिक coenzymes are also organic compound, but their association with the apoenzyme is only transient, usually occurring during the course of the catalysis. Furthermore, coenzymes serve as a cofactor in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example, coenzyme nicotine amide adenine dinucleotide, that is NAD, which you can remember by NAD, nicotine amide adenine dinucleotide, and NADP contains the vitamin niacin. A number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site, and at the same time form one or more coordination bond with the substrate. Example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Zinc is the cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. Now we are going to read summary in a speedy way. Although there is a bewildering diversity of living organisms, their chemical composition and metabolic reaction appears to be remarkably similar. The elemental composition of living tissue and non-living matter appear also to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen, and oxygen is higher in living system when compared to inanimate matter. The most abundant chemical in living organisms is water. There are thousands of small molecular weights, less than 1000 Dalton biomolecules, amino acids, monosaccharide and disaccharide, sugar, fatty acid, glycerol, nucleotide, nucleoside, and nitrogen bases are some of the organic compounds seen in living organisms. There are 20 types of amino acid and 5 types of nucleotides. Nucleotides are of 5 types. Fats and oils are glycerides, in which fatty acids are esterified to glycerol. Phospholipids contain in addition a phosphorylated nitrogenous compound. Only 3 types of macromolecule, that is protein, nucleic acid and polysaccharide, are found in living system. Protein, nucleic acid and polysaccharide. Protein, nucleic acid and polysaccharide are found in living system. Lipid, because of their association with the membrane, separate in the macromolecular fraction. Biomacromolecules are polymers. They are made of building blocks which are different. Protein are heteropolymer made of amino acid. Nucleic acid, RNA and DNA are composed of nucleotides. Biomacromolecules have a hierarchy of structures: primary, secondary, tertiary and quaternary. Nucleic acids serve as genetic material. Polysaccharides are components of cell walls in plant, fungi and also the exoskeletons of arthropods. They also are storage forms of energy. Example, starch and glycogen. Proteins serve a variety of cellular functions. Many of them are enzymes, some are antibodies, some are receptors, some are hormones and some are the structural proteins. Collagen is the most abundant protein in the animal world, and ribulose bisphosphate carboxylase oxygenase ribosco is the most abundant protein in the whole of the biosphere. Enzymes are protein which catalyze biochemical reaction in the cells. Enzymes are protein which catalyze biochemical reaction in the cells. Ribozymes are the nucleic acid with catalytic power. Proteinaceous enzymes exhibit substrate, especially require optimum temperature and pH for maximal activity. They are denatured at high temperature. Enzymes lower activation energy of reaction and enhance greatly the rate of reaction. Enzyme lowers activation energy of reaction and enhance greatly the rate of reaction. Nucleic acids carry hereditary information and are passed on from parental generation progeny. With this, we completed the chapter. Thank you for hearing. यहाँ तक टिके रहने के लिए आपका धन्यवाद. यानी आप बीच में से भी सुने हो स्किप करके, तो भी आपका थैंक यू.